In a genetics lab in Tel Aviv, a team of scientists did something no one had ever done before to one of the most important archaeological discoveries of the 20th century. They extracted DNA from the Dead Sea Scrolls, not from bones, not from teeth, from the parchment itself, the ancient animal skin that generations of scribes had written on more than 2,000 years ago, preserved by pure accident in the bone-dry caves above the Dead Sea for 2 millennia. And when the results came back, they didn't just confirm what historians already believed about these documents. They quietly rewrote part of the story, revealing that some of the most sacred fragments of scripture in human history may not belong together at all, that they came from animals of entirely different species, and possibly from entirely different places than anyone had assumed for the past 70 years. This is the story of that study, published in 2020 in one of the most respected scientific journals in the world, and what it actually found when researchers finally applied modern genetics to some of the oldest religious texts ever recovered. Let's start with why these scrolls matter so much in the first place, because the DNA results only make sense once you understand what was actually at stake. In the winter of 1946 or early 1947, the exact date is disputed even among the shepherds involved. A Bedouin teenager searching for a lost goat in the hills near the ancient settlement of Qumran, close to the northwestern shore of the Dead Sea, tossed a rock into a cave opening and heard something break. Investigating further, he discovered clay jars containing ancient scrolls of leather and papyrus, wrapped in linen, sealed away in the dry desert air, for what turned out to be roughly 2,000 years. That discovery kicked off nearly a decade of further searching, ultimately uncovering fragments from 11 different caves in the surrounding cliffs, totaling around 900 to 1,000 separate manuscripts, some substantial and largely intact, most reduced to small, brittle fragments, some no larger than a postage stamp. What made this discovery so extraordinary wasn't just its age, though the scrolls do include the oldest known surviving copies of the Hebrew Bible, some dating back as far as the 3rd century BCE, roughly 1,000 years older than the previously oldest known complete Hebrew biblical manuscripts. It was what the scrolls revealed about the actual text of Scripture itself. Before this discovery, scholars working on the Hebrew Bible had to rely on medieval copies, the oldest complete versions dating to around the 10th century CE. Meaning there was a gap of well over 1,000 years between when these texts were originally composed and the oldest surviving complete copies scholars had access to. The Dead Sea Scrolls closed a huge portion of that gap in one discovery, providing direct physical evidence of what these texts actually looked like centuries earlier than anyone had previously been able to verify. And remarkably, when compared against those much later medieval copies, the core text turned out to have been transmitted with an extraordinary degree of accuracy across 1,000 years of hand copying, a finding that itself reassured scholars about the reliability of textual transmission across that immense span of time. The scrolls include nearly complete copies of some biblical books, most famously an essentially intact copy of the book of Isaiah, alongside thousands of smaller fragments representing pieces of nearly every book in the Hebrew Bible, except the book of Esther, as well as numerous non-biblical religious texts, commentary, community rules, and other writings believed by most scholars to be connected to a Jewish sect active in the region during the Second Temple period, commonly associated with the Essenes, though that specific identification remains debated among historians. The community believed to have produced and preserved many of these scrolls appears, based on both the texts themselves and archaeological excavation of the nearby settlement at Qumran, to have lived a highly structured, disciplined communal life, organized around strict religious observance, ritual purity, and what several of the non-biblical scrolls describe as preparation for an anticipated final conflict between the forces of light and darkness. Some of these texts, including one known as the Community Rule, lay out detailed regulations for daily communal life, shared meals, property, and religious study, giving historians a rare, direct window into the actual internal life of a Jewish religious community roughly 2,000 years ago, written in their own words rather than filtered through the accounts of outside historians. Most scholars believe the scrolls were hidden in the surrounding caves deliberately, likely in the lead-up to or during the Jewish revolt against Rome that culminated in the destruction of the Second Temple in Jerusalem in 70 CE, an event the community may have anticipated as catastrophic enough to warrant physically hiding their sacred texts for safekeeping, quite possibly with the intention of one day returning to retrieve them, a return that, for reasons lost to history, never happened. The physical recovery and study of the scrolls themselves has its own complicated, occasionally troubled history, one that matters for understanding why the genetic study nearly 70 years later was such a difficult undertaking in the first place. Following their initial discovery, scrolls and fragments were sold, sometimes through antiquities dealers operating with limited transparency about origin or authenticity, and scattered across multiple institutions and private hands in the years before organized, systematic archaeological excavation of the Qumran caves began in earnest. Access to many of the fragments was, for decades, tightly restricted to a small, closed circle of scholars, a situation that generated significant frustration and controversy within the broader academic community until the 1990s, when photographic archives of the full collection were finally made widely available to researchers outside that original small group, dramatically accelerating scholarship on the scrolls. This messy, decades-long custodial history is directly relevant to understanding the genetic studies findings, since it explains both why fragments long assumed to belong together were grouped based on limited, sometimes rushed early analysis, and why modern human DNA contamination from so many different hands over so many decades became such an unavoidable obstacle for the 2020 research team. For over 70 years, one of the most difficult ongoing challenges in scroll research has been physically reconstructing which fragments actually belong together. For over 70 years, one of the most difficult ongoing challenges in scroll research has been physically reconstructing which fragments actually belong together. Thousands of individual scraps, damaged, faded, and scattered across multiple institutions and private collections around the world after decades of sale, trade, and sometimes questionable acquisition following the initial discoveries, needed to be matched and pieced back together based primarily on handwriting style, textual content, and physical characteristics like parchment color and thickness. This is painstaking, difficult work, and scholars have disagreed for decades about which fragments genuinely belong to the same original scroll versus which merely resemble each other by coincidence. This is exactly the problem the DNA research set out to help solve, and it approached it from an angle nobody had seriously attempted at this scale before. The scrolls, with only a small number of exceptions written on papyrus, were written primarily on parchment, a writing material made from prepared animal skin, most commonly sheep or goat in this specific region and era. That means every single scroll fragment is, in a very real sense, also a biological sample, a piece of the actual animal it was made from, sitting untouched in a climate-controlled desert cave for 2,000 years. In 2020, a research team led by neuroscientist and geneticist Oded Richavi at Tel Aviv University, working alongside biblical scholar Noam Mizrahi and a team of international collaborators, published their findings in the journal Cell, one of the most prestigious and rigorously peer-reviewed scientific journals in the world, describing an ambitious effort to extract and sequence ancient DNA directly from dozens of scroll fragments. Extracting usable ancient DNA from 2,000-year-old parchment is itself a genuinely difficult technical challenge, and it's worth understanding why, because it explains a lot about how careful and limited this kind of research has to be. Parchment production involves soaking, scraping, and stretching animal skin, then treating it with lime and other chemical processes to prepare it for writing, all of which degrades and damages any DNA present in the material far more severely than DNA preserved in bone or teeth typically is. On top of that, 2,000 years of storage, followed by decades of handling by researchers, museum conservators, and photographers under conditions that predate any modern understanding of DNA contamination control, means any genetic material recoverable from these fragments today exists in vanishingly small, highly fragmented quantities. Mixed together with genetic material from every person and environment the parchment has been exposed to since. Extracting anything usable at all required extremely sensitive, specialized laboratory techniques capable of detecting and sequencing extraordinarily degraded DNA fragments, working in dedicated ancient DNA cleanroom facilities specifically designed to minimize contamination. And even then, the research team had to develop rigorous statistical methods to distinguish genuine ancient signal from contamination and background noise, a technical achievement in its own right, independent of whatever historical conclusions the results ultimately supported. What they were hoping to find was straightforward in principle. If they could identify which specific animal, and ideally which specific species and even individual animal, each fragment's parchment came from, they could use genetic relationships to help confirm or challenge existing theories about which fragments belong to the same original scroll, since fragments from the same physical scroll should, logically, come from either the same animal or from animals raised and prepared together as part of the same production process. What they found surprised even the researchers who ran the study. The overwhelming majority of scroll fragments tested, as expected, came from sheep, consistent with what historians already understood about common parchment-making practices in the region during this period. But a small number of fragments came from an unexpected source entirely, cattle, a material far less commonly used for parchment in this specific era and region, and one that raised an immediate, genuinely interesting question. Cattle skin parchment production, and cattle husbandry generally, points toward different agricultural and geographic conditions than the arid sheep and goat herding environment immediately surrounding the Dead Sea and Qumran. This raised the real possibility that certain fragments, previously assumed without much question to have originated locally, near where they were eventually discovered, may actually have been produced somewhere else entirely, potentially somewhere with more fertile land suitable for cattle grazing, and then transported to the Qumran caves for storage or safekeeping. A genuinely different origin story than scholars had generally assumed. Even more significant than the species-level findings was what the genetic analysis revealed at a finer level of detail, comparing fragments that scholars had long assumed belonged to the same original scroll based purely on handwriting and textual content. In at least one notable case involving the Book of Jeremiah, fragments that had been grouped together for decades, treated in scholarly literature as pieces of a single original manuscript, turned out, upon genetic testing to come from genetically distinct sheep, animals different enough from each other that the research team concluded these fragments likely did not, in fact, originate from the same physical scroll after all, despite their textual and stylistic similarities. This was a genuinely significant finding for the field because it meant that at least some fragments long assumed by scholars to represent a single continuous document might actually represent multiple, separate copies of the same biblical text, produced independently, which has real implications for how scholars understand the diversity and possibly the standardization or lack thereof of biblical text production during this period. This detail matters more than it might initially seem, because the Book of Jeremiah, found among the Dead Sea Scrolls, has long been a subject of intense scholarly interest for an entirely separate reason. The version of Jeremiah preserved in some of these fragments differs meaningfully in length and content from the version of Jeremiah found in later, standardized Hebrew Bibles, more closely resembling instead the shorter version of the text preserved in the ancient Greek translation known as the Septuagint. This had already suggested to scholars, well before any genetic testing occurred, that multiple genuinely different versions of the Book of Jeremiah were circulating and being copied simultaneously during this period, rather than a single, fixed, universally agreed upon text. The genetic finding that some of these Jeremiah fragments came from entirely different animals adds a new physical layer of evidence supporting exactly that conclusion, suggesting these weren't simply damaged pieces of one original scroll, but potentially separate, independently produced manuscript traditions existing side by side, each reflecting a genuinely different textual lineage of the same biblical book, being copied and preserved in parallel by the same community. The study also uncovered something more subtle but genuinely fascinating about ancient scribal practice itself. Genetic analysis of the parchment suggested that scribes appear to have been deliberate and careful in selecting specific, high-quality animal skins for certain important texts, suggesting an intentional, considered production process rather than simply using whatever material happened to be available at the time, evidence of a level of care and craftsmanship in religious manuscript production that adds real texture to our understanding of how seriously these communities treated the physical creation of sacred texts, not just their content. It's worth being clear and honest about what this research did not find, because it would be easy and misleading to overstate the human element of this story. Despite hopes among some researchers that traces of human DNA might also be recoverable from the scrolls, perhaps from the scribe's own skin cells, sweat or oils left behind during the writing and handling process, the study found this human genetic signal was almost impossible to reliably isolate, and for an understandable reason. These scrolls have been extensively handled over decades by dozens, if not hundreds, of modern researchers, museum staff, and previous scholars since their discovery in the late 1940s and 1950s, long before anyone was thinking about DNA contamination protocols, since the entire field of ancient DNA analysis as a rigorous science barely existed at that time. That means any human genetic material recovered from the scrolls today is overwhelmingly likely to reflect modern contamination from the many people who have handled these fragments over the past 70-plus years, rather than genuine ancient DNA from the original scribes who wrote them 2,000 years ago. The research team was careful and transparent about this limitation in their published findings, which is exactly the kind of honest, self-critical reporting that distinguishes legitimate, peer-reviewed science from more sensational claims. This is actually a broader, well-documented problem across the entire field of ancient DNA research, not something specific to this particular study. Ancient DNA degrades over time into extremely short, fragmented pieces, and modern human DNA, shed constantly from skin and breath, is everywhere, on every surface, in every laboratory, making it exceptionally difficult to distinguish a handful of genuinely ancient human DNA fragments from the much larger, much more recent contamination introduced by literally anyone who has ever touched, breathed near, or handled a sample in the centuries or decades since its discovery. Serious ancient DNA laboratories now operate under extremely strict cleanroom protocols, specifically designed to minimize this problem. But for an artifact with as long and complicated a modern handling history as the Dead Sea Scrolls, some of which passed through the hands of antiquities dealers, private collectors and multiple research institutions across several countries before rigorous DNA-era protocols existed, that contamination problem is essentially unsolvable retroactively. The same fundamental challenge, separating genuine ancient signal from modern contamination, is exactly what pioneering ancient DNA researchers like Svante Pääbo had to solve decades earlier when his team first successfully sequenced Neanderthal DNA, work that eventually earned him the Nobel Prize in 2022. Pääbo's own early research was famously undermined for years by exactly this kind of contamination problem, with several of his initial DNA samples turning out, upon closer scrutiny, to actually be modern human DNA introduced by careless handling rather than genuine ancient genetic material, a humbling lesson that shaped an entire generation of far stricter laboratory protocols across the field. The Dead Sea Scrolls research team, working decades after those hard lessons were learned, was able to approach their own contamination problem with far more caution and skepticism than researchers in earlier decades might have applied, which is precisely why they were willing to be so transparent in their published findings about the limits of what their human DNA results could actually support. So, what the study actually delivered, and what makes it genuinely valuable to biblical scholarship rather than simply a curiosity, is real, useful data about the physical, biological origins of specific scroll fragments, information that's now being used by textual scholars to help refine long-standing debates about which fragments genuinely belong together, and to reconsider assumptions about where and how specific portions of these documents were originally produced. That's a genuinely significant contribution to a field that has spent 70 years piecing together fragments largely by eye and by scholarly judgment alone, now supplemented for the first time by an entirely independent, biologically grounded line of evidence. It's also worth sitting with what this discovery says about the broader relationship between ancient texts and modern science, because it's a genuinely unusual and elegant convergence. These are, after all, some of the most carefully studied religious documents in human history, examined for decades by paleographers, linguists, and historians, using every traditional tool available to them. And it took a completely different scientific discipline entirely, genetics, a field that didn't even exist as a formal science until the 20th century, to reveal something new about the animals these documents were physically made from. Information invisible to even the most careful traditional textual scholar working only with ink and handwriting, because it was never written down in any language at all. It was written instead into the genome of the animal itself, sitting quietly, unread, for 2,000 years, until molecular biology finally learned how to read that particular kind of text, too. That's ultimately what makes this story worth telling. Not a dramatic, world-shaking revelation that upends everything scholars thought they knew about Scripture, but something quieter and, in its own way, more remarkable. A genuinely rigorous scientific study, published in one of the most respected journals in the world, using real, careful genetic analysis to add a new physical layer of evidence to a field that has spent 7 decades working almost entirely with ink, parchment color, and careful eyesight. Some fragments long assumed to belong together may not. Some scrolls may have traveled further and from more varied places than anyone previously assumed. And somewhere in a lab in Tel Aviv, for the first time in 2,000 years, scientists read a message left behind not by a scribe, but by the sheep whose skin that scribe wrote on. A message it took modern genetics millennia to finally